Hi students, welcome back to this video. Listen to this. Failure does not mean game is over. It means try again with experience. Okay students, today's session we are going to study about these topics: artificial hybridization, double fertilization, embryo development, the development of endosperm, fruit, seed, then the two important phenomena which are found among plants, angiosperms, they are apomixis and polyembryony. Yes, let us study what is artificial hybridization. What is artificial hybridization? A plant breeder is interested in crossing different species and often different genera to combine desirable characters in order to produce superior varieties. So, actually, it's a crop improvement program in which desired pollen grains are used for pollination. Desired pollen grains are used for pollination. Then the important main two steps involved in this artificial pollination are: one is emasculation and the other one is bagging. First one is emasculation, second one is bagging. Then what do you mean by emasculation? Removal of anther from a bisexual flower. What is the purpose? To remove the anther in order to avoid self-pollination. After emasculation, this is emasculation, and after emasculation, the process is bagging. Here, emasculated flowers are covered with a suitable bag to prevent contamination of its stigma with the unwanted pollen grains. Then, when the stigma attains mature or receptivity, mature pollen grains collected from anther of male parent are dusted on the stigma and it is rebagged again. So that process is over artificial hybridization. What about the if the flower is a unisexual flower? Yes, there is no need of emasculation. If it is a unisexual flower, there is no need of emasculation. Female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Yes, next we are going to study about double fertilization. See, please go through this diagram. This is the diagram of a pistil or a carpel. The three important portions of the carpel is ovary. First one ovary, here it is ovary, and second one is style. Then third one is stigma. The three important parts are ovary, style, stigma. Here, now we are going to study about double fertilization. We have to concentrate this particular area, ovary. Inside the ovary, we can see ovule. So this portion is known as the ovule. Okay. So from the ovule, how the embryo sac development takes place, that we have studied. Then we have studied about the polar nuclei as well as the female gamete. So the fertilization takes place inside the embryo sac in angiosperm is double fertilization. So we can study what is double fertilization. Yes, pollen tube, when it enters into the synergids, or mainly the pollen tube, this portion is known as the pollen tube. Pollen tube enters into the embryo sac. Here we can see this particular portion is known as the egg. So here we have to put number 1, means first gamete, that is egg. And second one, near in the center we can see polar nuclei. So the fusion of one male gamete with this egg and the fusion of another male gamete with this polar nuclei is going to take place. Since there are two types of fertilization, it is known as double fertilization. So first one, we have to see what about the fusion of female egg with the male gamete. Okay, here in the double fertilization, there are two types of fertilization takes place. First, fertilization. Uh, as we all know, pollen grains consist of two male gametes. So one of the male gamete fuses with the egg and produce the zygote. This is the first fertilization. And the among these two male gamete, the other one male gamete fuses with the polar nuclei to produce primary endosperm nucleus. This primary endosperm nucleus later develop into endosperm. Then zygote gives rise to new plant, embryo, then embryo next gives rise to new plant. So next we can see what about the ploidy uh, of these structures. Means male gamete. What about the ploidy? Male gamete we have studied, it is haploid. Okay. Then what about the ploidy of the egg? That is also haploid. Then what is the resultant structure zygote? The ploidy is 2N. Then we have studied the ploidy of the polar nuclei is already 2N. Means since this reaches to the center from the each pole, the ploidy is 2N. What about the male gamete? We know that is haploid, and here it is diploid, 2N plus N. So what is the ploidy of the primary endosperm nuclei? 3N. From your examination point of view, it is very important. What about the ploidy of all these structures? Okay, children, once again, listen carefully. Male gamete is haploid. Female gamete is also haploid. So that the zygote is diploid. Then a male gamete is haploid. Then polar nuclei, the ploidy is 2N since they reaches to the center from each pole. Then 2N plus N, they combine to form 3N, primary endosperm nucleus. So this is known as double fertilization. Okay, you can see the diagrammatic sketch about the ploidy as well as double fertilization. Okay, here center polar nuclei. Here it is egg. One of the male gamete. Here you can see the two gametes. Two male gametes we can see. So this one is fuses with the egg to form the zygote. And the other male gamete fuses with this polar nuclei to form the primary endosperm nucleus. Okay, children, whatever is written here, it is already explained. Then, next is about, this is also we have explained, post-fertilization events. After fertilization, what all changes are there? Here you can see, based on this flow chart, you can see ovule gives rise to seeds, ovary gives rise to fruit. So based on this flow chart, it is very clear after fertilization, ovule gives rise into seeds and ovary gives rise to fruit. Then previously we have studied embryo is developed from the zygote, endosperm is developed from the primary endosperm nucleus. See here PEN primary endosperm nucleus, PEN. This gives rise to endosperm. Then here zygote zygote gives rise to embryo. So these are the post-fertilization events. Okay, primary endosperm nucleus gives rise to endosperm and zygote gives rise to embryo. Ovules gives rise to seeds and ovary develops into fruit. From your examination point of view, these are very important. So please copy down. Next is development of the embryo. Sorry, endosperm. Development of the endosperm. Primary endosperm cell divides repeatedly to form triploid endosperm. Endosperm cells are filled with the reserve food materials for the zygote. In common endosperm development, primary endosperm nucleus undergoes successive nuclear divisions to give free nuclei. This stage is called free nuclear endosperm. Number of free nuclei varies greatly. So endosperm becomes cellular due to cell wall formation. Here are some examples. Tender coconut water is a free nuclear endosperm made up of thousands of nuclei. And the surrounding white kernel, this portion, is the cellular endosperm. Yes, next is about the embryo development. This embryo is developed from the zygote. Zygote you can see here. So the zygote undergoes repeated divisions. First, pro-embryo is produced from that zygote. Later, it is changed into globular endosperm and heart-shaped endosperm. Finally, it changed into a mature embryo. So embryogeny means development of embryo. The word embryogeny refers to the development of embryo from the zygote. Then you have to study the structure of typical dicote embryo as well as the monocote embryo. See here, this is the dicote embryo and this is the structure of monocote embryo. Yes, mainly whatever it is, either dicote or monocote, it has an embryonal axis and cotyledons. In the case of dicote embryo, they possess two cotyledons. The two important parts of embryo are embryonal axis and cotyledons. In the case of dicote, there are two cotyledons present. Yes, here you can see a region named epicotyle region and hypocotyle region. So this is very clear. Hypocotyle region of the embryonal axis. What is this? The cylindrical portion below the level of cotyledons. Can you identify which is cotyledon? See, this cotyledon and this also cotyledon. So two cotyledons are present. Okay, this region is known as hypocotyle region. So what do you mean by hypocotyle region? The cylindrical portion below the level of cotyledon. Below the level of this cotyledon is known as hypocotyle region. That gives rise to uh, root tip or radicle. This gives rise to root tip or radicle. This hypocotyle gives rise to root tip or radicle. Then the portion above the embryonal axis, the portion of the embryonal axis above the cotyledon is known as epicotyle. This region is known as the epicotyle region. So that gives rise to plumule. Epicotyle region gives rise to plumule. This we have studied. The plumule gives rise to what? Later it develops into shoot system. What is this? Shoot system. Then what about the root radical gives rise to what? Yes, later this radical gives rise to root system. Okay, good. Radical later gives rise to root system, shoot system and root system. Okay, so what do you mean by epicotyl region? What do you mean by hypocotyl region? Which part is emerged from the epicotyl region and which portion is emerged from the hypocotyl region, etc. We studied. Then the root tip is covered by root cap, that we know already. Then what about the monocot embryo? Structure of monocot embryo. Actually, this is the structure of monocot embryo. You can see the diagram. Yes. Here it also possesses a cotyledon and embryonal axis. That cotyledon of the monocot seed is known as scutellum. Cotyledon of the monocot seed is known as scutellum. From your examination point of view, it is very important. What is scutellum? Okay, especially monocot plants, we know grass family. Then here embryonal axis, which is present above the cotyledon, that is epicotyle. Then below the cotyledon, that is known as hypocotyle, that is same what we studied in the dicot. But here one difference you can see. Coleorhiza, new terms you can see. One is coleorhiza and the other one is coleoptile. What do you mean by coleorhiza and coleoptile? Yes, the sheath which is present around the radicle, that is known as coleorhiza. The sheath which covers shoot system, that is known as coleoptile. These 2 points are also very important. Then next is about seed from ovule. We have studied after fertilization, ovule gives rise to seed. Here I should rub all this, or I should clear all drawings. What are the different parts we can see inside the seed? Yes, it consists of seed cord, cotyledons, and embryonal axis. The 3 important parts of a seed, either it is monocot or dicot, they are seed cord, cotyledons, listen, seed cord, cotyledons, and embryonal axis. Embryonal axis consists of, that we have studied, embryonal axis consists of plumule and radicle. Plumule gives rise to shoot system and radicle gives rise to root system. Okay. Yes, there are 2 types of seeds. When the seed matures, there are 2 types. They are, one is albuminous seed and the other one is non-albuminous seed. Or we can say endospermous seed or non-endospermous seed. So what is the word? Listen, endospermous seed, albuminous seed means endospermous seed, and here it is called non-endospermous seed. Non-endospermous seed. So 2 types. Non-albuminous seeds means non-endospermous seed. Albuminous seeds known as endospermous seed. What is the difference between these 2? Yes, when the endosperms are completely consumed by embryo, they are called non-endospermous seeds. Example, groundnut, beans, etc. Leguminous plants. Albuminous seeds or endospermous seed means, yes, they retain some endosperms. Wheat, maize, barley, rice, etc. are the examples. Yes, in some seeds, black pepper, wheat, etc., remnants of nucellus are present. So that is known as perisperm. You can see the perisperm in this pepper seed. This part is known as the perisperm. Actually, that is the persistent nucellus. What is nucellus? That you have studied while you studied about the structure of ovule. So please recollect it and try to understand. Then, integuments of ovules harden as tough protective seed coat. It has a small pore. The small pores which are present in the integument, they are known as micropyle. They are known as, what is it called? It is called as micropyle. What is the function of this micropyle? Through which carbon dioxide, water enter into the seed during germination. Okay, then what do you mean by seed dormancy? As the seed matures, it becomes dry by reducing water content 10 to 15 percentage moisture by mass. So that metabolic activity of the embryo slows down. It may enter a state of activity known as dormancy. Then under favorable conditions, moisture, oxygen and suitable temperature, they start to germinate. What are the advantages of seeds? Yes, seed formation is more dependable because pollination and fertilization are independent of water. Then they are dispersing into new habitats. It helps the species to colonize on that area. Then the seeds possess how many food reserves. Then next importance, hard seed coat protects the embryo. It is being the product of sexual reproduction. Yes, they generate new genetic combinations and variations. Then, dehydration and dormancy of mature seeds are crucial for storage of seeds. It can be used as food throughout the year and also to raise crop in the next season. These are the advantages or importance or significance of the seed. What about the viability of the seed? What do you mean by viability of seed? You can see so many plants here. Date palm is present here, that is Phoenix dactylifera. Then this is known as the Lupinus arcticus. Yes, they shows different viabilities, different seed viability. Few species, the seeds lose viability within a few months. Seeds of many species live for several years, but yes, some seeds can remain alive for hundreds of years. There are some seeds able to live for hundreds of years. Oldest is that of a lupin, Lupinus arcticus. This is the plant. Yes, it is found in Arctic tundra. So the seed germinated and flowered often after an estimated record 10,000 years of dormancy. Seed viability is 10,000. Then what about the date palm? Scientific name is Phoenix dactylifera. 2,000 years viability that shows. 10,000 is done by Lupinus arcticus and 2,000 years of viability found among date palm. Okay, this we have studied already. Or after fertilization, ovary gives rise to fruit. You can see the fruit here. The wall of the fruit is known as pericarp. This is the term, wall of the fruit. Please remember, wall of the fruit is known as pericarp. The fruits may be fleshy or dry. Dry fruit example, groundnut, mustard. Fleshy fruit example, guava, orange, mango, etc. Then, fruits are of two types, true fruits and false fruits. I should clear all drawings. Okay. What do you mean by true fruits? If the fruit develops only from ovary, they are known as true fruit. Example of most of the plants. The exceptions are there. They are known as false fruit. If the fruit developed from thalamus, this is the part. If the fruit is developed from thalamus, that is known as the false fruit. Do you know what are the examples of false fruit? Yes, apple, strawberry, and cashew. True fruit means fruits are developed from the ovary. Most of the fruits are true fruits. Exceptions, they are false fruit. False fruit are developed from thalamus. Example, cashew. This is the cashew. Then straw is there and here apple is there. Okay. Yes, this is a new term for you. What do you mean by parthenocarpic fruit? Formation of fruits without fertilization is known as parthenocarpic fruit. Example, banana and pineapple. Two examples are there. One is pineapple and banana. So what do you mean by parthenocarpy? Parthenocarpy means formation of fruits without fertilization. Yes, we can induce this parthenocarpy by applying growth hormones, plant hormones. So we can find in the case of this watermelon induced parthenocarpy means artificial parthenocarpy. This is done with the help of growth hormones. Natural parthenocarpy we can see in the case of banana as well as in the pineapple. Here you can see this is actually seeds, but these seeds are developed without fertilization. So these seeds are unable to grow. Okay, so formation of fruits without fertilization is known as parthenocarpy. We can induce parthenocarpy by using growth hormones. Okay, then the two terms, new terms or two phenomenons found among plants. This is not familiar to you. They are one is apomixis and the other one is polyembryony. Okay, what do you mean by apomixis and what do you mean by polyembryony? Yes, previously we have studied formation of fruit without fertilization is parthenocarpy. Here, formation of seeds without fertilization is known as apomixis. Production of seeds without fertilization is known as apomixis. Okay. Example, some species of astraceae and grasses. Okay. Then it's a form of asexual reproduction that mimics sexual reproduction. Why? Seeds are produced, so that mimics sexual reproduction. This is asexual because there is no fertilization. Yes, next we can see what is polyembryony. What do you mean by polyembryony? Many citrus and mango varieties. We can see occurrence of more than one embryo. Can you see here? See, multiple embryos are present, but only one develops into new plant. Why? Because zygotic embryo only gives rise to new plant, others all degenerate. Actually, what happened? In the nucellar tissues, some tissues differentiate into embryos, but these embryos will not grow into new plant later. Only the zygotic embryo will give rise to. Zygotic embryo means what? After fertilization, zygote is produced. The zygote gives rise to embryo. That embryo only will grow into new plant. But the nucellar embryos degenerate. Okay, so that process is known as polyembryony. Then, do you know advantages of this apomixis? It can be used for any other purpose. Yes, it is widely used in plant hybridization process. Yes, you can see if the wait. See, if the hybrids are made into apomictic, there is no segregation of characters in hybrid progeny. So farmers can keep on using hybrid seeds to raise new crop. Means usually after hybridization, plant hybridization, the seeds are unable to develop into new plant because it is artificially synthesized. If the seeds are made into apomictic, there is no segregation of characters in hybrid progeny. So farmers can keep on using hybrid seeds to raise new crop. Okay, students, what all things we have studied while we summarize? Please remember what all things we studied today. Yes, we have studied about starting with the double fertilization, artificial hybridization. Then next is yes about apomixis, polyembryony, about the seed dormancy, seed viability, etc. Okay, students, that's all about today's class. Thank you for watching this video. Thank you.